So let's continue. So we say, here is the basic picture, the structure of the bacteria. Then we talking about individually, one by one, and also their functions. Let's talking about the cell wall first. So we say cell wall, that's the special kind of structure for bacteria, not in the human being cells. So cell wall is the outside, we say most here, because some bacteria, like they only have cell wall, they don't have capsules. So that's for most bacteria, is the most outside the cover for the bacteria. And we also say some bacteria, they may have something outside of cell wall. That's including the capsules and some others. But for most bacteria, cell wall is the most outside covered. Outside covered. It's not good. So cell wall, we say it's located on the outside of the cytoplasmic membrane. Yeah, we say that membrane is similar like the human cells membrane. It's located outside it. So basic function of the cell wall, maintenance, and support the shape of the cell. So that's just like a cover in there. So that's the cell wall. So cell wall pretty simple. Cell wall pretty simple. Then we talking about the cytoplasmic membrane. Cytoplasmic membrane. This membrane pretty similar like a human being cell's membrane, but it located inside of cell wall. It composed the same structure we talked about before, like human being cells, is by two layers in there. Yeah, two layers in there. So basically, when we talk about human being cells, it looks like this structure in there. So these kind of structures, they have some important functions. The first functions we're talking about active transport some material to inside of cells. When we're talking about the human being cells and the material passing through the human being cells, we have positive way, we have active way. Active way, we're talking about it usually from the low concentrated part. For example, we're talking about some nutritions. Some nutritions is less nutrition in here. And that's like inside of cells. We need more nutrition, even we have a lot of nutrition in there, but we still need more nutrition. So that's active canal transport means use energy, use ATP. ATP is the form like living things can use directly, and use ATP transform nutrition from outside of cell memory to inside cell memory. So that happens in human being cells. It's pretty similar here. It's pretty similar here. They still need to use energy and transform from outside of the bacterial cell to inside of bacterial cell here. So when we talk about active, means we need to use energy. So that's anything go to inside of the bacterial, they must be passed through the memory part. And also they can generate some energy. We say they use some energy. And the cell memory, they have function, can generate some energy. So that's the second function there. And also they can form the cell wall. So they maintenance and form the cell wall. We say cell wall cover the plasma memory, but they need maintenance and forms by the plasma memory. When we talking about cell wall, we didn't talk about too much function. We just talking about the maintenance, the cell shape here. In here, we're talking about the bacterial cell shape. And another function, we say the secretion some enzyme and toxin. For the bacterial, that's some function of the bacterial, maintenance its life. But for the human being, this function certainly not good for the people. That's the mainly reason they can cause the people illness. So that's basically cytoplasma membrane. They have 4 important functions, but maybe not good for the human being. Then we move into the cytoplasm. We say outside we have cell wall first, and then we have cytoplasm membrane. It's the cover there, still cover there. And inside of these two covers is the cytoplasma. Yeah, cytoplasma. So cytoplasma is the liquid. It's the liquid or water. Yeah, you're just thinking about it's water. But inside of the cytoplasma, we also have something in there. Certainly, we still need to use microscope. Yeah, it's not like we directly can see it, use the eyes. We need to use microscope times thousands or even more. Yeah. And then we can see inside of the cytoplasma, we have something in there. So one part, it looks a little bit bigger, compared with the others. So this part pretty similar, like human being cells nucleus, but much simple than nucleus. They still have DNA. Yeah, in human being cells, in the inside the nucleus, we have axon. and inside the axon we have DNA. But for bacterial cells, they not that complicated. They just contain some DNA or maybe some other protein in there. So this area, we name it nucleate. Yeah, pretty similar like human being cells, nuclear. So that's a little bigger, yeah, little bigger. And we also have some smaller one. Yeah, we also have some smaller one. Plasmid, yeah, plasmid, yeah, plasmid. And also have some Robertsons. Yeah, also have some Robertsons. They not only have one, they have lot of plasmid, still have lot of. So basically, their maintenance like bacterial cells, like reproductions and metabolism and living. And certainly inside of cytoplasma, they even have some tiny small area. That's basically some nutrients and also some waste products, also inside of cytoplasma. That's like a small environment for the bacterial living. So that's generally in the cytoplasma. Then we're talking about structures of outside of cell wall. So we say cell wall for most bacteria, but for some bacteria, they have another thin kind of cover, and not for all the bacteria. So this kind of thin cover, we call it capsule. So the capsule, they have their functions. Yeah, they have their functions. And the bacteria with capsule, they will cause more toxins. So because they cover the bacteria, make the bacteria more stronger, yeah, more stronger, or even more bigger. So that's will limit it. We have a special kind of cells we talking about before, phagocytosis. Yeah, that kind of cell we also call eating cell. That's normally inside of a human body. We have a special kind of cells. It should be more bigger. Yeah, I didn't have space here. So basically, these kind of cells, if they find bacteria, they will cover it and then eat it and then digest it. So that's the special cells inside of our body, the way we kill the bacteria, kill the bacteria. So eating it and digest it. But when we say some special kind of bacteria, if they have capsules, it will be a little bit difficult for these kind of cells to eating. So means these kind of bacteria, if they have capsules, is more dangerous for people and easily got infections. It is more toxic. Yeah. So that's one function of the capsule. So the bacterial function is good for bacteria, certainly, but maybe not good for humans. So another functions, they have special identification functions. So these kind of identification functions may be good for the humans. So what means special kind of identification function? Means different bacteria, their capsules, their shape, their chemicals will be different. Like they have a special kind of like shape in there, or maybe special kind of protein in there, make it totally different with other bacteria. So like when we look at with microscope, we immediately find that special kind of bacteria. So these functions will be good for the, like we try to find a special kind of bacteria and kill it, because we immediately can find like what kind of bacteria is not good bacteria. We try to kill it with antibiotics. So like a special kind of antigen in there. So that's the second function. Basically, second function pretty similar with third function. We're talking about they have some special structure in there, and then we can identify, like we use for antigen, and we preventing the illness. We can immediately to use these kind of their functions to kill it. So that's the third one. And the last one, we're talking about special function for the capsules. They have a touching kind of functions. So means people have capsules, these kind of bacteria, they are pretty easily like attaching or sticker to the human's tissues. So that's another reason these kind of bacteria is easily cause the people infections. So still not good for the humans. But generally say for the capsules, for the bacteria, they have 4 different functions here. For the outside cell wall, first one we're talking about the capsule. And then we're talking about another two. The first one, right here. So this one we're talking about is look like a pretty long tail of the bacteria. Yeah, usually they're pretty long. So basically these structures, they located outside of the cell wall and even outside of the capsule if the bacteria have capsule. So mainly functions moving, like the leg of the bacteria. So that's basically they can pretty easily moving the bacteria to the nutrition area. What means nutrition for the bacteria? Human body cell. Yeah. They try to get nutrition in there. And also they try to touching some special kind of human body's cells or tissues. So they move in there and then they are touching there. So basically we're talking about a special word. Just means moving to special area. So that's it mainly functions Yeah, it's mainly functions. So thus, clinically, we have some example, like people got urination tract infections, like last semester. When we're talking about pathology, we're talking about urination infections. We're talking about some infections. They may happen in your bladder first. Yeah. Some bacteria pretty easily coming from outside the body and then go to bladder. And later on, some bacteria, they moving from bladder to the kidney. So that these kind of bacteria, usually they will have this kind of pretty easily moving kind of structure in there. So that's help the bacteria moving up from the urethra. That means this part to the bladder, and they also can moving from the bladder to the kidney. So that's this special kind of bacteria. So that's we say certainly these functions still not good for the human being. And also they have another functions, or maybe we can use because this kind of structure we still can use for identifications clinically. So that's after we identifications, we still can use for like help our immunity functions. We can use for specially antibody to against it. So immunity part we will talk about detail later. But generally say the first characters is not good for human being, but the second one certainly we can use for against it. So that's the first structures. Then we're talking about the second one. So second one, outside of cell wall is pretty shorter. Pretty similar like we looking the body hair, but certainly it's much shorter than body hair, because body hair we still can see with our eyes, but the bacterial hair we still need high times of the microscope. But compare like a bacterial size, look like a bacterial body hair, and it truly hair-like. So it is shorter, it's more shorter, and have more numbers usually. So these structures still important for bacteria are touching bacteria to human bodies cells, because they have this kind of body hair. So we say it's long kind of tail, mainly for moving. They still have some touching function, but mainly for moving. But the shorter one, mainly for touching. So that means the bacteria need touching the human body cells first, then they got infections. That's the first step, got infections. And also, in bacteria, they have special kind of pellets. Yes, special kind of pellets. We call it sex pellets. So sex pellets here is a little bit different when we're talking about sex functions, like in the human being. So generally say bacterial reproductions is not truly need like two different bacteria. Like usually we talk about human bodies cell like spores with eggs together, then they can produce the daughter's cells. But bacterial reproduction, we will talk about later, is totally different story. It can directly one bacteria, we say they have like a DNA kind of material in the center, and then they copy can make two DNA nucleotides first, and then these two it separate and form exactly the same two bacteria. Pretty similar, yeah, pretty similar. Like we're talking about human body cells division. Yeah, normally human body cells division is not sex cells division. It's different way. It's different way. So truly, like these bacterial reproductions, they didn't separate like female cells or male cells. But what's the sex pellets they working, and why we call it sex pellets? So that's just for some bacteria. They living way, they are living way, they are living together. Like we're talking about sometimes two bacteria they living together, and another two bacteria living together. So that's basically they connected each other by these kind of sex pellets. So that's basically the function of the pellet. So nothing benefit for the human being, but truly we can identify with microscope. We can identify some special kind of bacteria because they are living way, they always like two together. So that's the structure. Another structure we're talking about pili. Then we're talking about the bacterial spores. Yeah. So when we go back, bacterial structure here. So when we go back, I can use another one. Yeah, when we're talking about bacterial structure here, we say we can separate here. Yeah, we can separate here. So we say cell wall. Basically, for most bacteria, they will have cell wall. For the plasma membrane, like all the bacteria, they will be have plasma membrane. And the cytoplasma, all the bacteria will be have. And the nucleate, all the bacteria will be have. Robinson, every bacteria will be have. Plasmid, all the bacteria will be have. And outside of the cell wall, capsules, some bacteria will be have. Flagia, some bacteria will be high. And pili, I will be saying most bacteria will be high. Most bacteria will be high. So that's for basically bacteria. Yeah, basically bacteria. We only have some special kind of bacteria. Special kind of bacteria, or some of bacteria. They can form bacterial spores. Not all the bacteria, just some of them can form the bacterial spores, and in special kind of environments. So let's see what kind of bacteria and what kind of spores they will be forming. So now we're talking about the special kind of bacteria. They can form the spores. So spores, this look like a pretty thin kind of cover. A pretty thin kind of cover. And they just keep some DNA inside. Compared with bacteria, it's much simple. Yeah, much simple. So these kind of structures, so basically they can live in like for some pretty bad environments or conditions for a long term. So why these kind of bacteria? They can form the spores. And why? Yeah. So basically, it happens like the bacterial living environments, like nutrition is very poor, and they try to keep their living. They just keep the important living things like DNA in there, and form pretty stronger cover to protect it itself. So that's basically we're talking about the spores forming. So the spores, we say, mainly is DNA inside. Just a small amount cytoplasm. We say let it be water. Yeah, let it be water. Compare with bacteria, just let it be. And they cover with cell membrane. You can see two lines here inside, pretty thinner lines. Cell membrane is still in there. And very little water, just like a cytoplasm. And the most part is a big thin cover. Thin cover. You can see in here. So that's basically form a pretty stronger cover to protect it of the bacterial spores. So like they can against some heat and no water and even radiations, even chemicals. In other words, antibiotics. Yeah, chemical here. And some disinfectors. So they can against a lot of things to keep the bacteria living in spores form. Then, like we're talking about, when the environment suitable, like have more nutrition and the temperature suitable for bacterial growth, the spores can go back to bacterial form again, like cell capsules and cell wall, cell membranes, and more cytoplasma, more nucleus. So everything go back. Yeah, everything go back. So that's like during the bacterial spores stage, look like the bacterial sleep. In some special kind of condition, they can wake up. It's good for bacteria, but not good for humans. So these kind of bacteria, they usually cause the severe kind of illness and difficult to kill them. So that's a special kind of bacteria. They may have spores stage. So you can see this picture. So basically, we're talking about how the spore form. So that's originally the bacteria here. We still talking about we have cell wall outside of the cytoplasma membrane, and we have DNA. We have DNA, and the DNA will be copied. Yeah, we'll make a copy first, and then they will division to two cells. Yeah, to two cells. That's normally look like a normally form here. But later on, they form a different form or shape. One is bigger. Yeah, one is bigger. That's we call mother cells. Another one is smaller. Yeah, smaller. And this one will later on form the spores. We call pore spore. Yeah, pore spore. And then you can see, then you can see, like the mother cell losing some nutrition, but some baby cells, they got more cover or protection here. So that's more bigger cover, spores cover. And later on, finally, they will release. Yeah, they will release from originally bacterial cell from the individually spores. Yeah, individually spores. So that's individually spores. They can live in by itself. Yeah, it's living. It's just like sleep. Yeah, it's not diet. Yeah, it's not development. It's not make a copy. Yeah, even less metabolism. Yeah, just like sleep there. Yeah, just like sleep there. And later on environments suitable for their growing, they come back again. So that's basically for the spores and their forming. Then we talking about the cells growing circle or cells, how they're growing. We say the bacterial cell similar like body cells. Yeah, similar like body cells. It's one division to two, and two division to four, four division to eight. So pretty faster. Yeah, pretty faster. 1 to 2 and 2 to 4. So each one go to another 2. Yeah, each one go to another 2, another 2. So like originally one cell, after division, they will form 2 cells, then 4 cells, then 8 cells, then 16 cells. Yeah. So that's gradually coming. So that's the cells division. Yeah, cells division. So in this way, cells growing pretty faster. That's why it causes people illness pretty faster. So that's we say like naturally, yeah, naturally. But if we use like a calculation, yeah, calculate, like we put nutrition in there, like in our laboratory, and we try to calculate. So that's they may not follow that kind of code. They will like from start stage, they stay there, and later on they faster growing. But because environments, nutrition, and limited, they will stop there. And then later on they will die. So that's another story. Yeah, another story. So then we're talking about another conceptors. We say normal flora. Yeah, normal flora. What means normal flora? Yeah, normal flora. So we say whatever microorganism. Right now we're mainly talking about bacterial, and including others, yeah, including others microorganisms like fungi. So normally they're living everywhere. They're living everywhere, even in the superficial of the human being, like in the skin, inside the mouth and the nose, salt area, and the colon, and the vagina area. Almost have a lot of bacteria in there. Almost have bacteria. But normally, they are not cause the people illness. So these kind of bacteria, they keep some balance in there, not cause the illness. That's we call normal flora. Yeah, normal flora. 正常菌群 in Chinese. So we say not all the bacteria or microorganisms, they will cause the illness. Only some of them. Yeah, only some of them. So that's we call the normal flora. Other microorganisms, yeah, we're talking about bacteria and fungi here. We say we have some other microorganisms. They still live like in our environments on the superficial of body, like viruses and even some parasites. That belong to another group. That belong to another group. Even they still in there. But when we talking about normal flora, mainly we just talking about bacteria or maybe fungi. We are not especially talking about virus because virus reproduction rate is different with bacteria. And the parasite is different. That's another story. We're talking about separately later. But right now we're just talking about mainly bacteria. So we say not all the microorganisms will cause the illness. only some of them, they may cause the illness. So these kind of bacteria, we call it pathogen. So pathogen is the microorganism. We especially talking about the bacteria or virus if they cause the illness. Some bacteria, they may not cause the illness. They may even benefit for human beings. So that kind of microorganism, we are not call them pathogen. So remember, pathogen only if the bacteria cause the illness, then we call it pathogen. So we still go to a video to see the bacterial growth.
I can't hear anything anymore. Is that my problem?
It's coming.
Oh.
Can you see right now?
Bacterial growth. Bacteria are single-celled microbes. Bacteria reproduce by binary fission. In this process the bacterium, which is a single cell, divides into two identical daughter cells. Binary fission begins when the DNA of the bacterium divides into two replicates. The bacterial cell then elongates and splits into two daughter cells, each with identical DNA to the parent cell. Each daughter cell is a clone of the parent cell. When conditions are favorable, such as the right temperature and nutrients are available, some bacteria-like can divide every 20 minutes. This means that in just 7 hours one bacterium can generate 2,097,152 bacteria. After one more hour the number of bacteria will have risen to a colossal 16,777,216. That's why we can quickly become ill when pathogenic microbes invade our bodies. When grown in culture, a predictable pattern of growth in a bacterial population occurs. This pattern can be graphically represented as the number of living cells in a population over time and is known as a bacterial growth curve. Bacterial growth cycles in a growth curve consist of 4 phases: lag, exponential, stationary, and death. The bacterial growth curve represents the number of live cells in a bacterial population over a period of time. Lag phase. This initial phase is characterized by cellular activity but not growth. A small group of cells are placed in a nutrient-rich medium that allows them to synthesize proteins and other molecules necessary for replication. These cells increase in size, but no cell division occurs in the phase. Exponential phase. After the lag phase, bacterial cells enter the exponential phase. This is the time when the cells are dividing by binary fission and doubling in numbers after each generation time. Metabolic activity is high as DNA, RNA, cell wall components, and other substances necessary for growth are generated for division. It is in this growth phase that antibiotics and disinfectants are most effective, as these substances typically target bacteria cell walls or the protein synthesis processes of DNA transcription and RNA translation. Stationary phase. Eventually, the population growth experienced in the exponential phase begins to decline as the available nutrients become depleted and waste products start to accumulate. Bacterial cell growth reaches a plateau, or stationary phase, where the number of dividing cells equal the number of dying cells. This results in no overall population growth. Under the less favorable conditions, competition for nutrients increases and the cells become less metabolically active. Spore-forming bacteria produce endospores in this phase, and pathogenic bacteria begin to generate virulence factors that help them survive harsh conditions and consequently cause disease. Death phase. As nutrients become less available and waste products increase, the number of dying cells continues to rise. In the death phase, the number of living cells decreases exponentially and population growth experiences a sharp decline. As dying cells lyse or break open, they spill their contents into the environment, making these nutrients available to other bacteria. This helps spore-producing bacteria to survive long enough for spore production. Spores are able to survive the harsh conditions of the death phase and become growing bacteria when placed in an environment that supports life. Bacteria, like all living organisms, require an environment that is suitable for growth. This environment must meet several different factors that support bacterial growth. Such factors include oxygen, pH, temperature, and light requirements. Each of these factors may be different for different bacteria and limit the types of microbes that populate a particular environment. Bacteria can be categorized based on their oxygen requirement or tolerance levels. Bacteria that cannot survive without oxygen are known as obligate aerobes. These microbes are dependent upon oxygen, as they convert oxygen to energy during cellular respiration. Unlike bacteria that require oxygen, other bacteria cannot live in its presence. These microbes are called obligate anaerobes, and their metabolic processes for energy production are halted in the presence of oxygen. Other bacteria are facultative anaerobes and can grow with or without oxygen. In the absence of oxygen, they utilize either fermentation or anaerobic respiration for energy production. Aerotolerant anaerobes utilize anaerobic respiration but are not harmed in the presence of oxygen. Microaerophilic bacteria require oxygen but only grow where oxygen concentration levels are low. Campylobacter jejuni is an example of a microaerophilic bacterium that lives in the digestive tract of animals and is a major cause of foodborne illness in humans. Another important factor for bacterial growth is pH. Acidic environments have pH values that are less than 7, neutral environments have values at or near 7, and basic environments have pH values greater than 7. Bacteria that are acidophiles thrive in areas where the pH is less than 5, with an optimal growth value close to a pH of 3. These microbes can be found in locations such as hot springs and in the human body in acidic areas such as the vagina. The majority of bacteria grow best in sites with pH values close to 7. Temperature is another important factor for bacterial growth. Different bacteria grow at different adaptive temperatures. Some bacteria can adapt to temperatures below 4 degrees Celsius or even below 0 degrees Celsius. Generally, bacteria living in humans can adapt to temperatures of 20 to 45 degrees Celsius. Growth was best at 37 degrees Celsius or near body temperature. There are also bacteria that can withstand temperatures of 50 to 80 degrees Celsius, which can be found in hot springs and geothermal soils. Some bacteria require light for growth. These microbes have light-capturing pigments that are able to gather light energy at certain wavelengths and convert it to chemical energy. Cyanobacteria are examples of photoautotrophs that require light for photosynthesis. These microbes contain the pigment chlorophyll for light absorption and oxygen production through photosynthesis. Other bacteria, such as purple and green bacteria, do not produce oxygen and utilize sulfide or sulfur for photosynthesis. These bacteria contain bacteria chlorophyll, a pigment capable of absorbing shorter wavelengths of light than chlorophyll. Purple and green bacteria inhabit deep aquatic zones.
So that's word.
细菌是单细胞微生物，细菌通过二元裂变繁殖，在这个过程中，细菌一个单一的细胞。
Okay, my computer actually a little bit slow here. Okay, so let's come back. So that's we talking about the bacterial growth thing. And not all the bacteria will cause the illness. Only some of them cause the illness. Cause the illness bacteria, we call it pathogen. When bacteria cause the illness, or they try to cause the illness, as the human body try to against it or defending it. So for the human body, we have different way to against the bacterial infections. So some way is naturally kind of defenses. That's generally we say that's no special for it. What means no special for it? Means these kind of functions, they will against like almost all kind of microorganisms, not especially for special kind of organism. So that's including our skin, including our skin, or mucosa membrane, like inside the mouth or inside the nose. Skin cover pretty superficial of our body. So that's truly important part protected of our body against bacteria or other microorganism infections. So like normally we have lots of bacteria living in the superficial of the skin, but it does not cause the illness. If you cut your finger, you may cause the infections locally because your skin is broken. And then we're talking about another kind of cell, phagocytosis cell. So these kind of cells, just we're talking about like eating cells, eating cells. So they can cover the microorganism and digestion them. So that's another way we protected ourselves. And then we have some special kind of protein. We still can performing protection function. So that's belong to immunity systems part. We're talking about some detail later. So that's these three parts belong to naturally or no specially kind of protection functions. And then we have some specially kind of immunity function or acquired, means you get it later. So these kind of special kind of functions, usually they against special kind of microorganisms, like a special kind of bacterial infection or special kind of virus infection. So these part including the antibody and some lymph cells. We're still talking about some detail later. We just got some basic idea here. So then we're talking about some balance between the human beings, the function, and the microorganism infections. So microorganisms, we say including virus, including bacteria, including other microorganisms, they can cause the people illness. So that's depending like how toxic of the microorganisms and how stronger body immunity systems. So in other words, in TCM, we're talking about the body, how stronger defending T, 胃气 or 正气, yeah, and how stronger the toxins. If body 正气 is stronger, people will not go to ill. If toxin is much more toxin to against the body, people will go to ill. So that happens in the same passenger, like right now, COVID-19 evading, for some people may cause the severe kind of sick or even die. For another people, maybe just little be uncomfortable or maybe even no symptoms. So that's on the same toxin there, people's 正气 strong will not sick. People's 正气 weaker, people will get sick. So that's we're talking about same toxins. In other words, in other aspects, the people body 正气 the same. People body 正气 the same, like the same person. But got different microorganism infections. Some of them will cause people sick. Some of them maybe not. Like usually we can get some virus infections, the normal virus infections. Like some people maybe not got flu infections, but during the COVID-19 they got infections. So that's on the toxin part. Toxin strong is easily cause the people getting sick. Toxin is weaker. It's not that easy cause people sick. So that's totally is balanced things. So we say bacterial infections. Bacterial infections. Why they cause people illness? Basically by two aspects. One aspect is bacteria itself can invading the patients and cause the inflammations for the people. We learned inflammation last semester in our pathology class. We talked about that is locally some reactions like redness, swollen, painful, loose functions that can directly caused by bacterial infection locally. Another kind of cause patients illness is toxins. So we say some kind of bacteria, they have special kind of toxins, including some toxin inside of cell and some toxin outside of the cell. Inside of cells kind of toxin, when bacteria invading human body, they bring the toxins, still mainly go to locally. And outside kind of toxin, like we're talking about some bacterial cells, they can release some toxins to their environments. And these kind of toxins even can circulation with the blood go to other part of the body. So that's also another toxins for the human body. So, and also some structures, coming from the bacteria itself. Still can thinking about some toxins can release to the environments around the bacteria. So some toxin may be just a part of the cell wall. So that's they have different type of the bacteria and they release different kind of toxins. So we diagnose it. Like what kind of bacterial infections. We can go to library test. We can take some patient's blood, go to blood cultures to find bacteria, or maybe take something from salt air, go to culture. Culture means put on the special kind of no other microorganism living the nutrition air and waiting it grow up. So certainly we can take other sample coming from different part of the body depending what kind of illness, and then culture them. That's usually we can use in the library test. And certainly we have some other way we can find some antibody or maybe antigen directly from the blood circulations. That's for library tests we can find of the bacteria. So when bacterial infections, they need some nutrition. Like the reading we talking about some oxygen and some pH, and they can form there. And they try to protect it themselves, and they can move in, and then they protect it themselves. So they are touching locally, then they can live in there and then cause the illness. Later on, they cause the patient's symptoms. So during that time, the human body will have some reaction to it. That's still including our immunity. So, but generally we're talking about when bacteria invading the human being, they will be touching there first, then growing there, and then cause the symptoms. And then human body immunity systems will be reactions. And to see the balance, which side is more stronger than others. And when they use the drugs, medication or drugs, so basically we will control the bacterial cell wall to producing, and maybe we can kill some or eliminate some protein inside bacteria, or maybe we're working for the nucleic acid inside, or maybe cell memory. So for the drugs part, we're talking about some detail after our midterm. And the bacterial vaccine, thus they preventing the illness. Bacterial vaccine, they can including active and positive. So thus they still talking about some detail later. After we finish bacteria or virus or maybe some fungi and maybe some parasites, we especially talking about immunity. We discussed some basic idea. We have some positive or we have some active immunity functions. And sterilization and disinfection. That's the way we kill or maybe remove the microorganism. So that's maybe including the bacteria, maybe including the spores. So that's basically, if we're talking about the sterilizations, we will kill all the bacteria and the spores. But if we're talking about these infections, that's only kill most microorganisms, but they will not kill the spores. We know the spores is pretty hard to kill it, but sterilized, we can kill all the bacteria. So usually we use high pressure, high temperature. Then we can kill all the bacteria and the spores, like the needles we use, acupuncture needles. We must be sterilized. Others, like for the environment, we use these infections that still kill most microorganisms, but not the spores. But luckily, we don't have a lot of bacteria. They will have spores. So that's for today's